Guys, my name is Juan, and today I'm going to be talking to you about adaptive immunity, and specifically the role of B cells. Now, we are going to be going into a little bit of T cell function, but not too much. That'll be covered some other day. But for now, let's just really focus on B cells. And I'm going to be illustrating this in a really easy way. I'm going to be using Play-Doh, to kind of show you the structures of everything and what's going on. Adaptive immunity is one form of a couple of immune responses in the body. This type of immune response is the one that responds to diseases that we've already been in contact with. This is the reason that we can't be infected by the same disease twice. But how do the responses actually start? Well, first, we'll take a look at a few stages. Now, before we start, I want you guys to take a look at these. Now, you can see that most of the Play-Doh is actually on top of one another. Okay, in the cells, that's not really what it looks like. So now let's look at a couple of structures. We'll start with this one right here, the chief cell, the B cell. We'll worry about those little splotches on them later. B cells are made in the red bone marrow, hence the name B cell, or for bone. Now, B cells, their primary job is to look for an antigen, or antibody generator. These are found on foreign molecules. So we'll pretend for today that this green molecule right here is a bacteria. And it has these little orange things on them, and these are the antigens. When a B cell finds the antigen, it'll start to make these little guys right here called antibodies. Antibodies are proteins that the B cell makes so that it can attach itself to an antigen, that's shown right here. And it kind of sends a signal, and it shows the body what the cell is, what it looks like, and how to deal with it. Basically, it's just saying, hey, look over here. There's a bad cell, and here's how you fight it. Now, antibodies won't attach themselves to the entire cell, the entire antigen. They'll bind to these little things right here, the little white parts. These are called epitopes. An epitope is basically just an open surface on an antigen. Some antigens will only have one binding surface, this epitope, and then we have these. One antigen actually has two or more epitopes. We call these complete antigens. And what happens is you have more than one antibody that'll attach to it, and it'll send a signal to the rest of the body. When the body sees this kind of cell, it triggers a really huge and fast response. So it gets to do the things that it has to do a lot faster. Another type of antigen we can identify is called a hapten. It's basically a partial antigen because it has very limited epitope surface. So when an antibody does end up binding to it, it doesn't really trigger a response because it's not big enough, or it doesn't seem as dangerous. Now, before we go into what B cells do, we need to go into what T cells do. And we won't go into too much detail, but just know that T cells are some of the first guys on the job. Now, T cells have these little guys right here called T cell receptors. And what these receptors do is they find antigens that are floating around the body, bind to them, and tell the body to elicit many different responses. Now, once the T cell finds a pathogen, it will tell these cells, called a helper T cell, to go to a sensitized B cell, which we'll get to in a moment, and it'll release this kind of chemical. This chemical is called cytokine. A cytokine tells the B cell, okay, it's time to get all your processes going, and we need to fight this pathogen right now. Now, once the B cell has been told by the helper T cells that it's time to go fight these pathogens, it has bound to these antigens and will actually start to absorb them into the cell. Now, once the cell has actually vacuumed up these antigens, it'll actually put them back and present them on top of the cell through a process called endocytosis. It also has these little proteins right here on its surface called class 2 MHC proteins. And basically what they do is they help further the process of identifying the cell and knowing what it's supposed to do. Now, all of the processes that we have gone through to make the B cell activate and ready to go is called B cell activation. This is when the B cell starts to really kick in. Now, what they do is they divide into two different cells: plasma cells, memory B cells, both with different roles. Plasma cell is basically just a very beefy antibody generator, and it will continue to produce millions and millions of antibodies. Now, a memory B cell is a cell that doesn't really keep dividing, but is kept in more of a reserve. Now, if this cell were to ever see another pathogen that it has already been identified, what it will do is it will start to split into plasma cells. And now these plasma cells are going to completely overrun this pathogen, so that they don't even get a chance to do anything. And that's why if you ever feel sick and the next day you feel just fine, it's because of these cells. They've already seen this pathogen. They've already seen the type of disease that you have, so they can fight it much, much faster. Now, there are many types of antibodies. However, they're all split up into 5 different classes. Each one of these classes deals with antigens in a completely different way from one another. Now, we'll look at the first one. Now, let's say we have an intruder. These are our pathogens. And let's say this cell has these specific receptors. Now, for this pathogen to do its harm, it has to attach to these receptors and start to function the way it would. In this specific process, what happens instead is these antibodies will attach to the receptor and make it completely impossible for that pathogen to ever attach to that receptor. All of these pathogens will become completely useless. This process is called neutralization. Now let's look at another type of antibody response. Some antibodies can actually take and move antigens to bind to one if they seem to be a little bit too spread apart. You can see that this used to actually be 3 different pathogens, but they've been bound by the antibody. When the antigens are somewhat close, the antibody will actually just attach itself to both of them. So let's say there's an antigen that's really soluble, like a toxin. During this process, the complex structures are way too big and it's not soluble anymore. This is called precipitation. Now, if the antigens, rather than binding to a toxin, are binding to a bacteria or virus, this will actually be called agglutination. Once these foreign substances have been accumulated by all of the antibodies, they will trigger responses in the body to get rid of it. And now they're in a whole world of trouble. In another process called opsonization, the antibodies will completely cover an antigen and coat it like so. This will greatly enhance phagocytosis, which is basically just when a phagocyte, a different kind of immune cell, will actually eat the cell. Now, this happens through another process called adhesion, and you can see that the phagocyte actually attaches to the antigen in the entire cell. In the last reaction, sometimes the antibodies that attach to an antigen will actually change the shape of the antigen. Now you have these things right here in the brown called complement proteins that stick to this specific area and trigger the complement system. Basically, what the complement system does is it makes all the activities of the antibodies and the rest of the immune system much, much easier.
Now, the last thing we have to know about all of this is about inflammation. Inflammation is a response to a local injury. It can cause redness, heat, swelling, and pain. There are many different factors that can cause inflammation, something like direct impact, extreme temperature, or even infection. All of them can damage the surrounding tissue, they can kill other cells, and they have a bunch of other effects. Some of the cells that are damaged can release chemicals into the interstitial fluid, and because of the change in balance, a couple of things can happen. One of these effects is that the wound will be temporarily repaired, and no new pathogens can get inside. Inflammation will also slow down the spread of pathogens to other areas of the body. In the last response, there are many different immune defense systems that will be sent to this area, and they'll be able to overcome any pathogens and will promote permanent repairs, which is also called regeneration. In a nutshell, even though inflammation can be irritable and annoying, it's a good response. This means that the body is fighting pathogens and repairing everything the way it's supposed to be. All of these procedures will happen until the pathogen has been eliminated from the body. When the infection is cleared, the body will have produced antibodies to fight that specific pathogen, and if it infects you again, it will be completely destroyed quickly. And that's it for adaptive immunity and the role of B cells. Thank you for watching.